Yeah, what was the first law?
Law of
Law of
dominance.
dominance? Yes, what is it? What does it states? What does it say? How will you define that law?
Yeah, it states that in a heterozygous condition, the allele whose characters are expressed over the other allele are the dominant allele, and the characters of the dominant allele are called dominant characters, and the characters which appear in F1 generation.
Use the word trait, Maimuna, because character is height is a character. Shortness, short, dwarf, or tall is a trait of that character. Like skin color is a character. Fair or dark is the trait of that character. You understand?
Yes.
Eye color is a character. Blue, black, brown eyes are traits. Okay, continue.
Yeah. So in the F1 generation, dominant traits are found, and in the F2 generation, the recessive traits.
And how are you saying this F1 and F2? When the parents are pure line to begin with, right? Remember?
Yes.
This was the focal point of the last class, that in entrances, they can give you anything as a parent. So you just do not remember the line which is given in NCERT that in F1 generation, all are dominant. You have to also remember in F1, all are dominant and there is no recessive trait visible if the parents are pure line to begin with. Okay. Mamuna, is it clear?
Yes.
And then what we did, we also discussed something called incomplete dominance, remember? Something incomplete dominance, remember incomplete dominance?
Yes.
What was that?
I don't actually remember that.
Okay, so let me go to Madiha. Madiha, are there? Madiha is not there. Minashi,
Yes, sir.
remember what was incomplete dominance?
In incomplete dominance, the blending of traits takes place
Why?
because the dominant traits cannot be dominant over the recessive one.
Because yes, the dominant allele was not 100% dominant. It could not show its dominance in a heterozygous condition, correct?
Yes.
Yes. Then what happens? Blending happens. Does incomplete dominance affects the genotype?
No,
What does it affect then?
the affect phenotype.
Only the phenotype, right? Remember, Mamuna? Now are you recollecting it back?
Yes.
Yep. And we also discussed the example of snapdragon flower where incomplete dominance is seen. There are white flowers and there are red flowers. And when heterozygous condition arises, the flowers become pink, right? Because of blending in, correct? Yes or no?
Yes.
No. Okay, now let's come to, so I also told you that the second, so incomplete dominance is an exception of law of dominance, correct? So law of dominance is not a universal law. It has exceptions. A universal law is a law which is followed without exceptions. Okay, so the first law, law of dominance, is not a universal law. It has exceptions in nature. When Mendel gave this law, he was not aware of these exceptions. Later on, other scientists figured out that what Mendel told from pea plant experiment does not hold true for all other plants and animals. So the first exception, like law of incomplete dominance, came. There is another exception to it, which is called codominance, which happens in our blood grouping system, ABO blood grouping. So you know what is a blood group, everyone? So today we'll start with codominance. Now tell me, what is a blood group? Blood group, people, blood group. How do you, yeah, yes, Saifullah.
The antigen present in RBCs, right?
Yes, I told you to go back and read about ABO, right? Blood grouping. Even if you do not understand the codominancy part of it, just the blood grouping thing. You did that? You went back and studied blood group? So how, if I say if I say my blood group is O positive, okay? So my blood group is O positive. What does this means? Saifullah.
You don't have any antigen present on the RBC.
Okay, and what are these antigens?
Uh,
A and B, right?
yes.
Yeah, so there are two type of antigens present, antigen A and antigen B on RBC. Now, how many permutation combination can be there with blood grouping? If someone's blood group is O, let's start with the basic. Now you have to tell me all the possibilities with that, allele possibilities. If someone's blood group is A positive, then what could be the genotype, possible genotype? So this is the phenotype, right? A positive is the blood group, like the outcome. What could be the genotype possibilities? So mother and father both gave alleles, right? One possibility could be that the mother gave A antigen and the father also gave A antigen, right? Yes or no?
Yes, sir.
Let's keep the positive factor out for sake of simplicity for now. Let's say if it is A group. So A group means either this is possible, both mother and father gave A, yes? Or what is possible? Any other possibilities there?
IA and IO.
This I is not I, it is the representation for a chromosome, okay? We read it as IA, IO, but it's like I means the allele which came on that chromosome. So let's say mother gave A, one parent gave A and the other parent did not give any antigen. O means nil, negative, it's for 0. This is other possibility. Any other possibility that can happen to give a blood group?
No.
Are you sure, Madhya? Are you there, Madhya? You
Or
were...
like one parent gave A and the other one gave B.
Yes. So if one parent gave A and another one gave B, then what will be the blood group? You are saying it will be A? Now that is where the codominancy part comes in. So for A group, these are the only two possibilities. Similarly, for the B group, the two possibilities are either one parent gave B and the other also gave antigen B, or one parent gave antigen B and the other parent did not give any antigen. Is it clear? Everyone, Madhya, are you following?
Yes.
Yes, sir.
But in the case where one parent gave A and the other parent gave B, so normally if law of like Mendel's law were to be followed, then the trait is, sorry, the character is blood group, yes, and the traits are either A or B. And if Mendel's law of dominance was applied, then either A or B was dominant, right? Yes. But in this case, the blood group is neither A nor B. It's also not something in between. It is both A and B. So it is called AB group. You know about blood group A and B? So it means one parent gave antigen A, other gave antigen B, and both are equally expressing and equally dominant. So that's called codominance. You understand? Co means together, cooperation, co-education, okay? Co-curricular. Like together with your curriculum, you are doing some activities like you are doing theater, you are doing sports. That's called co-curricular activities, right? So together with A, B is expressing, and together with B, A is expressing. Both are expressing, so the blood group becomes AB. And this, when it was discovered in humans, it came out as an exception for Mendel's law of dominance, because one allele, this is a heterozygous state or not?
Yes, sir.
And in a heterozygous state, one allele should be dominant over the other. This is what Mendel told. But here,
Yeah.
in a heterozygous state, both the alleles are expressing equally. Okay. And this is called codominance. You understood? everyone?
Yes, sir.
Now for AB to happen, there is only one condition, that both the antigens come from both the parents. And if, what is then O group? O group is the most, like, it's not recessive, it is absence. Do not think it is recessive because it is nothing. So from first parent, there was no antigen that came on RBC1. And from other parent also, there was no antigen. And there is only one possibility, right, to make a O blood group. Yes or no?
Yes,
Often
sir.
students and teachers get a little confused, thinking that in this case, this is also, is this a heterozygous condition?
Yes.
Yes.
No, it is not a heterozygous condition, because a heterozygous condition is where two different alleles are present. In this, only one allele is present. On the other place, there is no allele for the antigen. Do you understand?
Yes, sir.
It is like a heterozygous condition, but it is not a heterozygous condition. Do you understand, everyone? So we often
Yes, sir.
think that A is dominant over O, which is equivalent to saying that, 1 is dominant over 0. Of course, if there is 1 and there is 0, the 1 will be there, right? 0 is nothing. Dominancy can only be said to exist when two things are present and one is dominant over the other, right? If only one thing is present, then can it be dominant over the thing which is not present? Yes or no?
Yes, sir.
If two fighters are fighting, you can say that, oh, fighter A is dominant, boxer A is dominant over boxer B, right? If in the ring there is only one boxer, can you say that boxer A is dominant over the empty space?
No.
No, right? So this is what the condition is. In this group, only one parent gave an antigen, other parent did not give an antigen. So the blood group O is absence of both A and B antigen. Right now, blood group O is absence of both A and B antigen. But I have seen teachers also teaching it like this, and students also remembering it like this, that A and B are dominant over O, but O is nothing. Okay, everyone. So in human blood group, Madhya, are you following? Is it easier? Is it difficult?
No, I am following. It's easy.
Easy, right? So in human blood grouping, the A and B antigen, write down, antigen A and B are co-dominant with each other. Both expressed in a heterozygous condition. Antigen A and B are co-dominant with each other. Both are produced, both are produced, both are expressed, and antigens are like, they are present on the surface of the RBCs like markers. Okay, so they are molecules which are present on the surface of RBCs. So both are expressed equally in each other's presence. Both are expressed equally in each other's presence. Okay, now coming to the level of allele. Now this was about the antigen. Okay, so there are two antigens, A and B. Or what are these antigens? Basically, they are sugar molecules. Is it clear, everyone?
Yes, sir.
Yes or no? They are sugar molecules. Now at the level of alleles, so there are two antigens but three alleles. So here, this part IO, there is a allele for this. So there has to be something, no? That gene, remember I told you that if one chromosome, there is something present at this spot, in the father's chromosome, something has to be present at this spot, right? Both of them will be allele pairing. Yes. So this cannot be vacant. There is no antigen, but there is a allele. So write down there are 3 alleles. The allele for A antigen, the allele for A antigen, allele for B antigen, and allele for, and there is one allele which do not give rise to any antigen. So we call it empty allele or I allele, small I. Okay. At the level of allele, both A and A antigen allele both this is dominant over the empty allele. Is it clear? Because write down this I allele does not code for any sugar. That's why there is no antigen produced. Is it clear, Madhya? Are you following?
Yes, sir.
So if one parent on the chromosome gives this allele, the small I allele, then it is an empty allele. Empty means it will not code for any sugar, so no antigen will be produced. So nothing will be at the surface of the RBC. Is it clear? The surface of the RBC will be empty, not empty, but it will be lacking both A and B from this parent. Is it clear? And if both parent provided this allele, then the surface of the RBC will be devoid of both A and B antigen. Okay. But whenever the allele of A antigen or allele of B antigen is present along with I allele, they both are dominant and they will be produced because there is nothing to, compete with them. Is it clear? So the point of dominancy only makes sense for the allelic at the allelic level. Madhya, is it clear?
Yes, sir.
But not at the expressed antigen level because it is nothing, it's nil. Okay. Is it clear to everyone? Madhya, Saifullah, Minashi, Mamuna,
Yes, sir.
Azia? Everyone is on the same page? Now, what kind of questions, yes, Mazia saying yes, can come? So you understand, write down first, write down. The gene for antigen of blood, the gene for antigen in some textbook it is written as gene I. It is just to make you understand. It's not known as gene I, but NCERT uses the term gene I. You can write it down. The gene for the antigen, gene I, has 3 alleles. Allele for antigen A, first allele for antigen A, which is also in NCERT regarded as, so in the beginning, who was reading it as IA? Was it Madhya or Mamuna or Minashi? Who was it? Someone said IAIB, right? This IA means the allele for antigen A, okay? The version of the gene for antigen A. Is it clear, everyone? Am I audible, people?
Yes, sir.
Yes, sir.
Yeah. Then allele for antigen B, and then an empty allele which does not code for any sugar, which is called I, small I. So these are the 3 alleles of the gene I. So these 3 alleles, write down. IA gives rise, IA and IB, write it like this. IA and IB give rise to slightly different sugars. Slightly different sugars. Okay, everyone? Sorry. These slightly different sugars give rise to antigen A and antigen B. So antigen A and antigen B both are sugars. The difference is they are just slightly different in their structure and signatures. So they are called antigen A and antigen B. Whereas the third allele, small I, small I, does not produce any sugar. Does not produce any sugar. So there is no antigen. Clear, Madhya?
Yes, sir.
So write down at the allelic level. Allele IA and IB are dominant over. At the allelic level, allele IA and IB are dominant over small I, which means they express. But of course they will only express because the other allele is not showing anything. But next line is important. But when both the alleles, when both IA and IB alleles are present together, but when both alleles IA and IB are present together, they both express their own sugars. In the bracket, write antigen. They both express their own sugars. Okay? Clear, people? Am I clear?
Yes, sir.
This is because of codominancy. This is because of codominancy of these two alleles. Okay. Now, the type of questions that comes. Can we go to the question for codominancy? Would you like to solve? Now here, the point of a genotypic ratio or a phenotypic ratio also comes in, and also along with the blood group. So write down the first question. Everyone, write down question number 1. You have to solve it and give me the answer in chat, okay? Everyone is clear? Shall I begin with the first question, testing your concept, or anyone has a doubt?
Yes, sir.
Yes, write down. Parent A parent A has a blood group A. Parent A has a blood group A. Okay. I'm starting with one of the complicated ones. And parent B has a blood group B. Okay. Some of their kids had blood group had some of their kids had blood group O. You understood the question, everyone? Parent A has a blood group, parent B had a B blood group, and some of their kids had O blood groups. Now you have to tell me the first part. Is this possible? Is it a possible scenario? Is this scenario correct or false? You have to tell me this. Second, you have to tell me if possible, then what are the genotypes of parent A and parent B? Okay, A and B. And what will be the genotypic and phenotypic ratio in generation 1? This is F1, this is parent. Is it clear, everyone? The question is clear to all of you? Madhiya, is it clear?
Yes,
There are
sir.
two parents. One parent had blood group O. The question is giving you phenotypes only. One parent had blood group A and one parent had blood group B. And there is a statement made that some of the kids had blood group O. Now, first you have to tell me, is this possible? If it is possible, yes or no. If it is not possible, then there is no point of going to question 2 and 3, right? But if it is possible, what is the genotype of A and B? Even if it is not possible, what is the genotype of A and B? So first and second, you have to find anyways. Third only depends when it is possible. So find and tell me. You just do the question. I will come to someone for the answers. Okay. I'll come to different people for different sections of the question. I'm giving you 2 minutes, everyone. Time starts now. Okay. Madhiya, Saifullah, Minashi. Clear? The question is clear. Are you doing it? Mamuna. Mamuna, are you there?
Yes, sir.
Yeah, please do it. Giving you 2 minutes. Those who will finish the question and find the answers will just write done in the chat and I'll come to them. Okay, understood, everyone? Everyone, am I audible?
Yes, sir.
Yes, just write done in the chat if you are done with, if you have solved the question. Now you have to be a little faster. Okay, try to be faster with every problem. Okay, Vajay, you are done? Yes. But yeah, was it easy?
The first and the second part was easy. The third part also, the genotypic ratio is easy, but the phenotypic one.
Okay, but at least you are not struggling with genetics till now, right? I think you will not struggle with genetics. I think you just have not given it a proper consideration while studying. It is a very, very technically sound part of biology. So if you understand the concepts, you can just solve it. And remember, you are the benchmark of the batch, right, Madhya?
Yes, sir.
You specifically had a problem with this topic, but okay, most of you are done. Who's not done? 1, 2, 3, 4. Try to finish faster, everyone. Just give me one. I'll just be back in some time. Okay, everyone is done. Okay, finally, good. Am I audible? So I will ask Madhya to lead me. Okay, Madhya? Let's solve the answer. And let's see everyone has done the same thing or not. So first tell me if parent A's blood group is A and the other parent's blood group is B. And it is given that some kids have blood group O. First thing, is it possible?
Yes.
Yes, it is possible. And if it is possible, then what will be the genotype of parent A?
I A.
Yes.
I A, and it will be I A and I O.
See, there are two possibilities for A blood group. You are right. IA and IA and IO.
IO.
Right?
Yes.
And there are two possibilities for B group, which is IB and IBIO. But if some kids have blood group O, what is the only possibility that can give rise to IO? Sorry,
IO.
So if this is the only possibility, then can the parents be this?
The parents will not have IA and IB group.
If any parent is homozygous for either A or B, is it possible that in the next generation you can have a O blood group?
No.
No, right? Everyone understands? If I say one parent is this, IA, and other parent is IB, IO. So is there a possibility then that in the next generation any kid can be O blood group?
No.
No, because this parent will always give either A or A. Even if this parents give O, the blood group will be A only, right? To have a blood group O, you need at least one allele of O, which is no antigen on both the parents. You understand, everyone?
Yes, sir.
Amana, is it clear?
Yes, sir.
So this is not possible. This and this is not possible, right? So what could be the only possible genotype of parent A, IAIO and IBIO? Then only some kids can get O from here and O from here and become IOIO, right? So yes, this is possible. Everyone understood the first part? Say yes or no.
Yes.
Second part is also solved, right? If possible, this is the only possibility, right? Let's come to the third part, GR and PR in F1. Now again, what we do to solve GR and PR? We make Punnett square. Now, what are the possible gametes that parent A can form? Either IA or IO. Yes? Yes or no?
Yes.
Parent B can make either IB or IO, right? What could be the 4 kids? This will be IA.
IA, IB, IA, IO.
Yes. And
IB, IO, and IO.
so what is the genotypic ratio? What are the possible genotypes? First, tell me that. One is this one, IA, IB, right? Is 2. What is the other possibility?
IA, IO.
IA, IO is 2. What is the other possibility?
IB, IO.
IB, IO is 2. What is the other possibility?
IO.
IO, IO. So what is the genotypic ratio?
It's 1:1:1:1:2.
Everyone understood? Sorry. 1:1:1:1. Everyone got the same answer? Madhya,
Yes.
Mamuna, Minashi, Mazia, got the same answer?
Yes, sir.
Yes, very good. Now let's go to phenotypic ratio. Now, what are the possible blood groups is the phenotypic ratio? What are the possibilities? Either a blood group can be A, or it can be B, or it can be AB, or it can be O, right? Are all 4 present? What will be the blood group of this first person, first kid?
AB.
Here the blood group will be AB. Here
A.
A. Here
B.
and here
O.
So what is the phenotypic ratio also?
It's also 1:2. 1:2.
Is it clear, everyone?
Sir.
Everyone got the same answer?
Yes.
Right?
Yes, sir.
Very good. So which means you understand this. So I'll just give you one more question which has come in boards and then we'll move forward. Write down the question. Parent A has a blood group B. Parent A has a blood group B and is heterozygous for the alleles. And is heterozygous for the alleles.
Sir, can you repeat from the start?
Parent A has a blood group B. Okay. Okay. Is it clear, Saifullah? Parent A
Yes,
has
sir.
blood group B and is heterozygous for its alleles. This is the information given to you in the question. Parent B has a blood group O. Parent B has a blood group O. Find the phenotypic and genotypic ratio. Find the phenotypic and genotypic ratio in first generation. This is the only information given to you. Very less information. Parent A has blood group B and is heterozygous for the alleles, and parent B has blood group O. No information for the alleles. You have to find GR and PR. Can you do it, everyone? Yes, do it. Please write done when you are done with the answers. Okay. Madhya is enjoying, right? Solving these.
Yeah.
Yes. That's good. Okay. Saiful is done as well. Very good. So the reason why I'm making you do this in front of me, because I just do not want to give you the concepts and move ahead. That will not tell me if you have understood the concept, you have also learned to use it to find the right answer. So it becomes important to do certain questions in the class only. I will give you many such questions in an assignment also. Quickly finish up. Are you there, Mazia? Am I audible to you? Okay, I think everyone is done. No. Mazia, you're there? Are you solving it? Yes. Please let me know when you're finished. Are you stuck somewhere? Would you like to do it with me? Okay, you're doing. Please finish. Others, did you find the question easy or hard? Once you understand, it's easy, no?
Easy.
Easy peasy. It's easy for Madhya. I'm very glad. Okay, so I would ask Mamuna to help me. Mamuna, I'll follow your lead, okay? Are you there?
Yes, sir.
Yes, so let's start with the figuring out the genotype for parent A. What is the genotype for parent A?
IB and
IB.
IO.
IO, right? because
Right.
it's written that it is B group and heterozygous. So this is the only possibility, correct? Very good. What is the for B? Parent B?
IO.
Yes. So there is no need to give the information about the allele, right? Combination. If it is O group, it has to be IO and IO, correct?
Yes.
Yes, perfect. Now you have to, then it's simple. You have to tell me the genotypic and phenotypic ratio in the first generation. So tell me, what should I write here?
IB and IO.
IB and IO, and here
IO.
IO and IO. Okay, then what will be the first?
IBIO.
IB, IO,
IBIO.
IB, IO, yes,
IO.
okay,
And IO.
okay. Now what is the genotypic ratio?
Genotypic ratio is 1:1. So it's IBIO and
Genotypic
IO.
ratio is not 1:1. Genotypic ratio is 2:2.
Yeah, sorry.
You cannot cancel it out and make it simple, okay? Because
Yes.
there are total 4 offsprings. It's not like 2 and 2 we take cancel and it becomes 1:1. It's ratio, okay?
Yes.
Yeah. So genotypic ratio is 2:2. And what are the genotypes?
IBIO and IO.
Perfect. And what is the phenotypic ratio?
2:2.
Yes. And what are the possible phenotypes, blood groups?
B, a blood group, or O.
Oh, there is no possibility of A here, right?
Yes.
So now you understood that. So these are the questions that can be asked in boards. Now in entrances, they just ask you questions like, they will just make a scenario. So the scenario is the kid's blood group is A, okay, and the one of the parents' blood group is B, and the other parent's blood group is O. Okay, everyone. The kid is likely to be first their real biological child, second adopted child, third can't say, fourth information is not enough, like something like that.
Adopted.
Yes, you understood why? Because from real biological parents, one with blood group B, other with blood group O, it is not possible to have a kid with a blood group A. Yes. But if the question says that one parent had blood group AB and the other parent had blood group O, is it possible to have the kid with blood group B? A, B and O are the two parents. Can the kid have blood group B, biological kid?
Yes.
Yes, it is possible, right? Do everyone understands what kind of questions can be asked?
Yes, sir.
Yes. Okay, very good. So the codominancy part and incomplete dominancy part both are clear to all of you, no doubts, right? Shall I move forward now to the second law we are going to start today? First law and its two exceptions are clear. Yes. Now let's go to second law. Write down law of segregation. Now write down about, anyone knows what is law of segregation? Any idea? People? Any idea what is law of segregation?
Yes, sir.
Yes, please tell me.
It means that when the two traits come together in one hybrid pair, so both the characters will not mix. I mean, the traits will not mix each other, and the gametes also receive one of the two alleles during the meiosis of the chromosome.
Yes, perfect. So what Mamuna said in slightly complicated manner, I'm just going to tell you very simply. Law of segregation simply says it does not matter that both of the alleles coming from both the parents are same or different. At the time of gamete formation, they will be segregated without blending. Is it clear? Actually, throughout this time, we were following law of segregation to understand law of dominance. Let me go back and tell you where we were segregating things. Let's start from the very beginning. In law of dominance, I told you, this is one parent, right? Parent A. People, just pay attention here. Yes. And this is parent B, right? Yes or no?
Yes,
This
sir.
allele is dominant over this allele. Capital T is dominant over small t. But when we were making gametes, did it ever affected it? In gamete formation, always one allele independently got segregated. Yes or no? Gametes, law of segregation in very simple terms says gametes are always pure. Even in the case of incomplete dominance, let me come to incomplete dominance now. Yes. In incomplete dominance, this hybrid was pink, right? Yes or no?
Yes.
So there was blending of traits at the level of the phenotype. Yes or no? Yes or no?
Yes.
But even when even when this capital T and small t, considering that there was an incomplete dominance, even when this heterozygous got made gametes, the gametes were pure again, right? Gametes did not give rise to pink character at all, even if there was an incomplete dominance, even if there was dominance, even if there was codominance. In all the cases, when you make gametes, whatever was giving rise to the phenotype, the two alleles together, they always segregate independently, right? Yes or no?
Yes.
At the level of gametes, there is no blending ever. The blending only happens at the level of the zygote and the embryo and when the phenotype appears, not at the level of gametes. And law of segregation is the only law which is universally accepted and universal law. So write down law of segregation is also known as law of segregation is also known as law of purity of gametes. Law of segregation is also known as law of purity of gametes. It is the only universal law among the three, with no exception. It is a universal law without any exception. Is it clear, everyone? You understand?
Yes, sir.
Now write down this law. Sorry. This law states that alleles do not show any blending. Alleles do not show any blending at the level of gametes. At the level of gametes. And both the traits, so for example, capital T is bringing tall trait, small t is bringing short trait, but both these traits are recovered as such like as it is without any blending. Both the traits are recovered as such in the F2 generation. You understand? So when meiosis happens and half of the chromosome goes one side, half of the chromosome goes other side to make gametes, these two alleles goes independent, like say they become separate without any blending. So what I mean to say is, let's say IA, because this is a recent one, you can understand, IA and IB, when they were present together, what was the blood group? AB, right?
AB.
Because what is happening in the case of blood grouping? Codominance, correct?
Yes, sir.
When capital R and small r were present together, what was the flower color? Pink. Incomplete dominance happening, correct? When capital T, small t is present, both the alleles are present together, what is the height of the plant?
Tall.
Tall, because in this case we do not see any, it is just follows the rule of dominance. In all these three cases, somewhere it is codominant, somewhere it is incomplete dominance, somewhere the, and all three are heterozygous conditions, yes or no?
Yes.
But when gametes will be formed, what gametes here? It will be a separate pure IA gamete and a separate pure IB gamete, right? Yes or no? And
Yes,
this
sir.
IA, if it again gets a chance to be present with IO, what will be the blood group? A group. You understand? So the same allele which was present with B giving rise to AB, the same allele when it is present in the next generation with O gives rise to A group. So do you think that anything got affected because of their presence with something? No, right? They at their gamete level stay pure. Nothing affected them. Do you understand? The same allele can show a different phenotype with a different allele, and the same allele can show a different phenotype with a different allele. Is it making sense?
Yes.
And why does it happen? Because at the level of gamete formation, at the level of meiosis, the law of segregation is playing a role. It is the universal law without an exception. Never ever it happens that your alleles get affected by the other allele in its presence. Yes, they both together will express differently, but when they are making gametes again, they are pure as alleles, not affected by the other partner whatsoever. Is it clear? Here also, capital R, small r present together give rise to pink. Now, when gametes will be formed, there will be one gamete with R and the other gamete with R, right? These two will be the gametes. Yes.
Yes, sir.
And in the next generation, if this R gets a chance to be with another capital R, what will be the phenotype? Red. Yes. So
Yes, sir.
do you think this blending because of incomplete dominance affected this capital R allele? No, because when gametes were formed, again it became pure one, which can give rise to anything based on the combination. Okay, everyone.
Yes, sir.
Yep. Understood law of segregation? Everyone understood the law of segregation? Or is there any doubt? Any doubt?
No, sir.
Okay. Now write down third law. Okay, before going to third law, I have to tell you about, so the third law is called law of independent assortment. Second law does not have any exception, so you just have to understand the law. Third law is called law of independent assortment. Now, this law also has exceptions, so it is not a universal law, but let's first figure out what is the law. As the law says, it is a law of independent assortment, but independent assortment of what? independent assortment of characters. Is it clear?
Yes.
Till now, we have only dealt with a single character which is having multiple alleles. Is it clear? But we only dealt with one character, whether it is height, whether it is flower color, whether it is blood group, but it is just one character with multiple alleles. Yes. In case of blood group, there are 3 alleles. In case of other, there are 2 alleles. But the character is single. But sometimes, in reality, we do not talk about just one character. No, there are multiple characters. So let's say there are 2 characters present. If I say that a pea plant parent A has round and wrinkled seed, what does it mean on a genetic level? What does this term means? What does round and wrinkled seed? Now come back to the first page. Is round seed a dominant character or a recessive character?
Dominant.
So many a times this will be given to you in the question paper, that if they are talking about they will tell you whether it's dominant or recessive. Am I audible, everyone? Am I audible and visible?
Yes.
Yes, I think I lost the connection in between, but I'm back. Okay. Is the screen also visible to you, the screen sharing?
Yes.
Yeah. Okay, I was asking, if I say there is a round and wrinkled seed, then how many characters I'm talking about?
2.
Yes, this one word is talking about which character? Seed shape, right? Sorry, round and wrinkled are wrong statement. Round and yellow seeds. So this one character is talking about seed shape, and this is talking about seed
Color.
color, right? Yeah. Can you see there are 2 characters that we are dealing with simultaneously? Okay. These are called so to find this is called a multi-character problem, and for that we have to deal with dihybrid cross. Till now, whatever crosses we have made was called monohybrid cross, because there were one character each having multiple alleles. Now there are multiple characters having multiple alleles. So now tell me whether round is a dominant trait or recessive trait.
Dominant.
Round seed shape. If it is round, it is dominant trait. Yes.
Yes.
And yellow color is
Recessive.
seed color yellow. Is it recessive?
Dominant.
Sorry,
Dominant.
dominant. Who said recessive? It's not recessive. It's go back to the the chart which we made in the beginning. For seed color, what was the dominant trait? Yellow, right? That's where I told you we get green seeds at our home, right? Green pea. That is a recessive trait that we propagate, farmers propagate. Is it clear? Green seed color is a recessive trait. And with seed shape, round is the dominant trait. So both are dominant here. Now tell me, for round and yellow, what are the possible genotypes? For a round seed, what could be the possible genotypes? One is capital R, right? Homozygous. And
Yes,
because
sir.
it is a dominant trait, if it is capital R and small r, that will also be a round, right? Yes or no?
Yes, sir.
Okay. Sometimes I have seen questions in entrances. NCERT talks about round in terms of R, but I've seen question in entrances where they will, these two will be denoted as capital W, and small w, sorry, capital W and small w. You know why? I discussed this in the last class. Actually, we give the letter with respect to the recessive character. In the seed shape, what is the recessive character? Wrinkled. And wrinkled word starts with W. So for dominant, which is round, we write it as capital W. Is it clear, everyone? So if you find this in entrance, and don't get confused, this is the right connotation. This is the right way to write. Okay, correct way to write. This is the way which makes it easier to understand. So NCERT follows this way. I will also follow this way for the rest of my class. But is it clear that you do not have to get confused with this?
Yes, sir.
Yes.
Anyone who's finding it confusing, please tell me. So these are the two possible genotypes for round seed. What are the possible genotypes for yellow seed? Take Y. Capital Y, right? Or capital Y, small y, correct? Yes or no? Everyone?
Yes, sir.
Now tell me, the first point here is, if I'm saying I have a plant which is pure line round and pure line yellow, a plant with pure line round seeds, round and yellow seeds. So what will be the genotype? How will I write it? Pure line round and pure line yellow seeds. What will be the genotype? Madhya?
It will be capital R, and capital E.
Capital Y, right? And if I cross it with a pure line wrinkled and pure line green seeds, then I'm talking about parent generation. Pure line wrinkled and pure line green. Madhya?
Small r, and small g.
Yes, so let me use the same color coding. Make things easier. Okay. So for this, you are saying capital R, capital R, capital Y, crossed with, for this you are saying, what is the genotype?
Smaller,
Small r, small r.
and small g, small g. A
Why?
small y, small y.
Yes, you don't change the alphabet, right? Small y, right? So can anyone tell me if this is the parent generation, how will be made the Punnett square now? First tell me how many gametes. To do this, you have to tell me. First step is, oh sorry, I'll use a neutral color now. What are the gametes possible? How many different type of gametes are possible from this genotype?
4.
4. Which one?
R, R, Y, Y. Capital R.
No. But you are making a classical mistake here. Remember, this is one character and this is another character. Yes. It's a multi-character problem now. If you're saying in one gamete only R will go, then only the information for seed shape is there. There is no information for seed color, right? There has to have an information for seed color as well. Do you understand? When characters are more, in each gamete, there will be one copy of every character, no information about every character. Otherwise, this gamete just has the information for seed shape. What about seed color? So seed color will also be there. RY will be one gamete, right? What will be the other gamete? Sorry?
RY.
The same, right?
Sir, your voice is breaking.
So for pure line hybrid parents, there is only one type of gamete possible. Am I audible now? Hello, am I audible, everyone? Madiha, Saifullah, Minashi.
Yes, sir.
Yes,
Am I
sir.
audible?
Sir.
Audible and visible, right? Clearly, no lag.
Yes, sir.
So with a pure line hybrid parent containing 2 characters and 2 alleles for each character. So for round pure line, capital R is there. And for yellow pure line, capital Y is there. When these parents will make gametes, in the gamete, there will be information for both the characters. Actually, in our body, there are more than 25,000 characters. And for each character, there is an allele which comes in 23 pairs of chromosome. So we are just dealing with 2 dihybrid cross here. So this will be the only gamete possible because the other gamete possible will also be the same one, right? So both are the same types, yes or no? This R and this Y and this R and this Y. Do you understand? This is how we make gametes. Saifullah, you understand?
Yes, sir.
So both the gametes are technically same, right? So I can just say that there is only one type of gamete possible. Yes?
Yes, sir.
Same is with here. This R and this Y will become RY, right? One gamete. And this R and this Y will become the other gamete. whatever permutation combination I make, because they are pure line, this will be the only type of gamete possible. Yes or no? You understand, everyone?
Yes, sir.
Yep. So if I cross these two parents, then what will be the genotype and phenotype of the first generation? Tell me.
Capital R, small r, capital Y, small y.
Yes. All the progenies will be this only, correct? Yes or no?
Yes.
In F1 generation, all the progenies will be this, because there are only two type of gametes made which can be crossed now. Is it clear, everyone? So this is the only genotype. And what is the only phenotype? All of them will be
Round and ring, round and yellow.
and yellow. Why all of them will be round and yellow? Because in the first generation, like go to the first law, first cross, monohybrid cross. In the first generation, only the dominants one show. I think this is what someone mentioned in the last class, right? I think Reem did. She's not present today. Okay, so capital R, small r, capital Y, small y is round and yellow. Yes. Now, if we self cross, this is the tricky part now. Now you have to do this and tell me. If we self cross F1 to find F2, what will be the genotypic ratio and phenotypic ratio in F2? Can you find out, everyone? Take some time and find out. This is the question. Can you do this? Would you like to try first before I solve it? Madhya,
Yes, sir.
would you like to do this?
Yes.
Basically, what I'm asking you to do is, I'm asking you to do this. Capital R, capital Y, crossed with capital R, small r, capital Y, small y. This is parent 1 and this is parent 2. So first make the possible gametes, then put it in the columns and then make the Punnett square. Are you doing it, everyone? I'm giving you 5 minutes to all of you. Okay? Say yes or no. Confirmation, please.
Yes, sir.
Yep. Do it. You have to tell me the GR and PR of the F2. We have not come to the law yet. I will tell you what the law is. First, we are learning a dihybrid cross. This is called inheritance of two genes that we are studying. Those who will complete will write done in the chat. And if anyone is stuck, just write down that got stuck or need help. Just stop there. Anyone who's done? Okay, so some of you are done. For others, okay, Madhya has done. Okay, so I'll ask Saifullah to lead me this time. Saifullah, are there?
Yes, sir.
Yes, please. Let's start. Okay. So you tell me first, what are the possible gametes that this parent can make? Parent one.
Capital R, capital Y.
One is this gamete possible, having both the information for both the characters, and both are homozygous, right? Like, sorry, and both are dominant one. Then
Capital R, small y.
yes, capital R small y, the other combination, yes, and
Small r, capital Y.
small r capital Y and
Small r, small y.
small r small y, right? These are the four combinations possible from this. Any other permutation combination possible for gametes?
No.
And if both of them are same, we are self-crossing, the same four will be possible for this parent as well, right?
Yes, sir.
Yes. Everyone understood till here? So this time when we will make a Punnett square, it's not going to be a 4 boxed one. Instead, how many boxes will be there? 4 gametes on one side and 4 gametes on other. You understand, everyone? Yes. So on this side, we will write parents A's gamete. What are the gametes? Capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y, right, Saifullah? And on this side,
Yes, sir.
the same, right?
Yes.
So the trick is if you can make the gametes properly, rest everything is just a unit square. Now tell me, what will be the first offspring? possible?
Capital R, capital R, capital Y, capital Y.
Yes. So when we are making it from gametes, when we are making a zygote, when they are fusing and making an organism, we write both the alleles for the same character together, for the same gene together. Okay. So the two alleles for round here, like the seed shape here, from one parent it is capital R, which is round, and for other parent also it is capital R, which is round. So we write both of them together. In this case,
Capital R, capital R, capital Y, small y.
yes. Continue. Keep telling me.
Capital R, small r, capital Y, capital Y.
Okay.
Capital R, small r, capital Y, small y.
Okay.
Capital R, capital R, capital Y, small y.
Yes.
Capital R, small y, small y.
Yes.
Capital R, small r, capital Y, small y.
Everyone is following? Capital R. Next.
Capital R, small y, small y.
Yes. Here.
Capital R, small r, capital Y, capital Y.
Yes. So similarly, we will keep filling in every box. I'm just sparing Saifullah from repeating this thing. Here it will be small r, right? Capital Y,
Yes,
capital Y.
capital Y.
Yes.
Small r, capital Y, small y.
So you're seeing that all different permutation combinations are making up. Just by including one more character to the inheritance, it got so complex. If I include 3, then what will happen?
It will be more complex.
But will there be more squares?
Yeah,
No.
no.
No. Okay, we'll see why and why not. So capital R, small r here, and capital
Capital
Y
Y, small y.
here. Capital R.
Capital R, small y, small y.
Okay.
Small r, capital Y, small y.
Yes.
Small r, small y, small y.
So everyone, this is the Punnett square that we made. Now first tell me, what were the parents? These were the parents. I'm writing the parents again here. Okay. For this cross, the parents were capital R, capital Y, small y, right? This was the parent into the same because it was a self cross, right?
Yes, sir.
How many of these 16 are like the parent? Exactly. How many genotypes are exactly like the parent? Can anyone count and tell me quickly?
Four.
4.
4, right? And if I ask you, where are they? They are present right here on the diagonal, right? Can you see this, everyone?
Yes, sir.
These 4 are like the parents out of 16, which means 12 are not like the parents. Yes. So talking about first the genotypic ratio, we are clear that now there are more genotypes possible. Now tell me, there is a trick to figure it out. So whenever you make a dihybrid cross, and if you started, so this is the F2 generation, right? So in F1 generation, all of them were hybrid for both the characters, and all of them were round and wrinkled, whatever is the dominant character. The recessive one only is visible in the F2 here also. And what is the genotype? What are the different possible genotypes? See, this is one, capital R, capital Y, small y, right? Do you see anything like this anywhere else?
No.
No. So this is unique. Let's come to second. Do you see anything like this anywhere else? Yes, there is something like
Yes.
this right here. These two are same, right? Any other genotype which is same? Can you find out anywhere in the cross? Yes? Or no?
Capital R, small y is common.
So this is, but this is different, no? Capital R, small y is different. And then these two are same, right? These two are same. Do you understand?
Yes.
Then these four are same. Similarly, these two are same. This is unique. These two are same, and this is unique, right? So there's a diagonal relationship here. Can you all figure out? So the ratio will be 1 is to 2 is to this 1 is to these two is to these four together is to these two again is to 1. These two are done. This one is done. Is to these two and this one. So it is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So it first increases 1 2 1 2, then 4 2 1 2 1. Total, can you add how many are there? How many genotypes are there? 1 2 3 4 5 6 7 8 9 genotypes are there, and total offsprings are 16. If you add them up, it will be 16, right? Yes, everyone, you understand? This is the genotypic ratio in F2 in a dihybrid cross. And what do you think is the phenotypic ratio? Now this is interesting. Tell me. Let's figure it out. For the first one, what will this be? Round and yellow, right? Safula, you're there. You have to lead me in this. Is the genotypic ratio clear to you? There's
Yes,
a
sir.
diagonal relationship. Always go through the diagonals. In the phenotypic, what will this be? Round and yellow?
Yes.
So I'm writing round and yellow. What will this be?
Round and yellow.
Again, so I'm writing round yellow. This
Round yellow.
round yellow. This
Also round yellow.
round yellow. This one
Round yellow.
round yellow. This one,
Round green.
yes, round green. First RG. This one
Round yellow.
round yellow. This one
Round green.
round green. Second RG. This one
Round yellow.
round yellow. This one,
Round yellow.
yes, round yellow. This one,
Wrinkled yellow.
first one, wrinkled yellow. This one,
Wrinkled yellow.
yes, second wrinkled yellow. This one,
Round yellow.
round yellow. This one,
Round green.
third round green, third RG, right? This one,
Wrinkled yellow.
third wrinkled yellow. This one,
Wrinkled green.
wrinkled green. First. So if you see the phenotypic ratio, either it could be round yellow is to, it could be round green is to, it could be wrinkled yellow is to, it could be wrinkled green. Yes, everyone? And the ratio will be, how many round yellow are there? Count it.
9.
9. How many wrinkled green are there?
3.
Which one? Wrinkled green, 1, 2, and 3, right?
Yes.
How many wrinkled yellow? 1, 2,
3.
and 3. Yes. And how many wrinkled green?
1.
1. So phenotypic ratio will be 9:3:3:1. Is it clear, everyone? In F2, this is the genotypic and phenotypic ratio in a dihybrid cross. Is it clear?
Yes, sir.
Now, dihybrid crosses are slightly, you have to pay more attention to it when you are making it, but there are certain shorthand tricks that once you get to understand this, towards the end I will give you all those tricks in hand notes, but you have to understand what are we doing fundamentally first. But do you realize that we started with pure line parents in the first generation, right? The parent generation, sorry, in the parent generation it was all pure line, right, for the dominant and for the recessive both, correct?
Yes,
Then
sir.
we got hybrids in the genotypically hybrid in the F1, but all were dominant in terms of phenotype, right? And from round and yellow parents, which we self-crossed, in F2, we got a wrinkled and green. This character was not there in any of the parent before, right? Was it there in parent or F1? In none of the generation, wrinkled and green was there. Understand? Sorry, in parent generation, there was wrinkled and green, like to begin with. Here, this was wrinkled and green. So, but in for F1 generation, there was no wrinkled or no green. But in F2, again, there is wrinkled and green character visible. Do you understand? Yes or no? Is it clear?
Yes.
Yes,
Now
sir.
coming back to law of independent assortment, write down what does this law. So everyone understood what do we mean? What do we mean by dihybrid cross? Dihybrid cross is clear.
Yes, sir.
Towards the end of the chapter, I will also make you. I will teach you tri-hybrid cross, where 3 characters will be taken just for the entrance purposes, not for boards. But write down law of independent assortment. Law of independent assortment states that when two pairs of characters are combined in a hybrid plant, when two pair of characters are combined in a hybrid plant, one pair was for seed color and the other pair was for the seed shape, right? You understand? So when two pair of characters are combined in a hybrid plant, the segregation of one pair of characters, is independent of the other pair of characters.
Sir, can you repeat from hybrid plant?
Yes. The law states that when two pair of characters are combined in a hybrid plant or animal, so don't write plant, just write in a hybrid or in a hybrid, just in a hybrid. When two pairs of characters are combined in a hybrid, the segregation of one pair of characters is independent of the segregation of other pair of characters. Okay, the segregation of one pair of characters is independent of the segregation of other pair of characters, which simply means here, look here. These are two pairs, right? In a hybrid, one pair of character and the other pair of character, both are in hybrid. Yes? Yes or no?
Yes, sir.
And it also means that this was supposed to be round when present like this, right? In a hybrid setup, this was supposed to be round and this was supposed to be yellow. But this pair got segregated when you make gametes and when it is when the combinations come back again in a zygote, the round and yellow gets segregated independent from each other. No character influences the segregation of other character is what this law says. That if round and yellow are from one parent, if round and yellow are coming, it is not true that in every child round and yellow will go together. Do you understand? In some kids, there will be round and green also. In some kids, there will be something which was not present in the parents, wrinkled and green also. In some kids, there will be wrinkled and yellow also. Do you understand? This is what law of independent assortment says. That round and yellow as a character, two hybrid together, will get segregated independent. If they were dependent on each other, then they will go together in every generation, but that does not happen. No two characters are dependent on each other and they segregate separately when they go into the next generation. And this is where all different permutation combinations are formed. This is where variations come in sexual reproduction. Are you understanding, everyone? What parents were, kids are different from the parents because of law of independent assortment of characters. Is it clear, everyone? Say yes or no?
Yes, sir.
Yes. Now this law, write down, law of independent assortment has some exceptions. So the exception which is there is called linkage. Linkage. So the exception to law of independent assortment is linkage. It is not a universal law, as I told you. Linkage and recombination. This I will start in the next class because it's a very big topic. And along with it, I will also start mutations. We will take both of them together, like not together, but one after the other, but we'll finish it in the next class. So this is your homework. You have to go back and read this. Okay, everyone? Linkage and recombination and mutation. This is an exception. Linkage is an exception to third law. Okay. Now simply what does linkage says? What Mendel did an experiment on pea plant and he's figured out that it does not matter which two characters I am studying. He studied actually all different characters. He studied seed shape with seed color. He studied height with flower color, height with the flower position, flower color with the seed color, everything. And he realized never ever two characters were passing on together. Always they were getting segregated, and in kids there was other kind of permutation combinations appearing. Do you understand, everyone? Is it understandable, people? There was always something which parents did not had. Independent assortment was always happening. Is it clear to you, Madiha?
Yes, sir.
Yes. So he gave a theory, he gave a law that there is independent assortment of characters which takes place all the time. At that time, Mendel did not know, or he did not did the same study in other model systems. So that was the limitation of Mendel law. Mendel's law are not incorrect. They are limited because he only did experiment in pea plant. But later on, scientists realized that what he said about characters is also not true. It's not universal. There are exceptions. Like law of dominance is always not true. There is codominance in our blood group. There is incomplete dominance in Snapdragon. Similarly, law of independent assortment is also not true. There are certain characters which are linked together, and they always go to an offspring together. They do not get segregated at the time of gamete formation. Do you understand?
Yes,
Do you
sir.
understand, everyone? Yes. So those two genes which together go from one generation to another are called linked genes. They have a linkage in between. So it is like 1+1 offer. If you want to get gene A, you will get gene B with it because they are both linked together. And this is an exception to independent assortment. Those genes do not assort independently. They always go together. Do you understand, everyone? So that is linkage. And what is recombination? We'll discuss it in the next class. So please read before you come to the next class and try making some more dihybrid crosses. Let me give you some as homework. Try making a dihybrid cross for this. So write down the homework. Hybrid tall with round cross to homozygous short with hybrid round. Okay, I want a dihybrid cross, the genotypic and phenotypic ratio for all these. Okay, in the next class. Second, let's say wrinkled with hybrid yellow cross to wrinkled with. Just do these two to figure out whether you can make the. So they are the parents. You have to cross them and tell me in the next generation what is the genotypic ratio and phenotypic ratio. Is it clear, everyone? The homework?